Well, it was a really wonderful introduction, and I'm not sure what to say, except I'm really grateful to be here. I'm grateful to the rector and the deans and the entire faculty of SES for the award you've given me. Being named honoris causa is a very, very meaningful thing for me. And I really want to thank all of you for the trust and the honor that you've given me. What I want to talk about today is a synopsis of work that's going on at Stanford right now in the area of open science. It's not just at Stanford, though, it's all over the world, but I want to give you a flavor for our take on it and how some of the technologies that you've already heard a little bit about are contributing to, I think, a new way of thinking about making data more available, making data more useful, and hopefully expanding the ways that we can make discovery from the data that we achieve in our everyday scientific research. What I want to do is, first of all, just give you a flavor for what we're talking about. I realize this is a somewhat eclectic audience. I guess most of you are computer scientists, but many of you come from throughout the SES enterprise. Let's just talk about this particular kind of business that I'm involved in. I'm involved in a business where there's absolutely intense competition, more competition than you could ever imagine to achieve product, and where that achievement of results really depends on trade secrets. But the problem is, no one can really trust the results unless we know what the trade secrets are. And the products that are generated as a consequence basically may be flawed if those trade secrets themselves have problems. And the customers, meaning the people who pay for the work, the people who pay for research, obviously want to make sure that their money is well invested, that the work being done is legitimate, and they want to make sure that ultimately those trade secrets are made open so that other people can engage in the same kind of work. And so basically, it's a very curious kind of business where in order to be successful, you have to be very secretive. But in order to be extraordinarily successful, you have to be very generous and very open. And the verification of the products that are delivered need to be verified as advertised, which means understanding what those trade secrets are. And the development of related products by competitors is really what everybody is interested in. And yet, obviously, that may not necessarily be in the interest of the original people who are involved in the work. And this probably sounds like no business that you've ever studied in this particular faculty, but it's one that we deal with all the time. And it's the business of science. And in the business of science, we're sort of sensitive to the fact that everything we do is governed by, well, government. That in Europe, if we want to get support from Horizon Europe, we need to make a commitment to making our results available, our methods available. We have to make sure that everything we do is open to other investigators. In the US, we're seeing the same kind of thing. The Office of Science and Technology Policy of the White House recently issued an order saying all federal support for research has to ultimately result in open science, open access to publication, open access to data. And my God, are people scared about that idea. In the United States, the National Institutes of Health, where I get most of my funding, recently issued a data sharing policy which requires everyone receiving NIH support to share his or her data and his or her methods, his or her software. This is a really big deal in the United States, where we have an entrepreneurial philosophy behind a lot of what scientists do, and suddenly they can no longer be isolated and siloed, but have to make their work available to others in ways which is, in some sense, very uncomfortable to them. This is why the world is changing. And it's changing because these changes are allowing things that were never possible before. Let me tell you a story. I want to tell you a story about a faculty member who works with me at Stanford, who's in my division, a guy whose name is Pravesh Khatri. Pravesh is a guy who's made remarkable scientific discoveries, and I'll tell you about those in a moment. Pravesh calls himself a data parasite. And he's actually very proud of the fact that he calls himself that. Pravesh has made remarkable discoveries. He usually looks at data that come out of what are called DNA microarrays. These are experiments where a robot has put on a chip every gene in the body, if you will, And he can run other people will run experiments looking and seeing how these genes get turned on and turned off over time, and use this information about the activation and inactivation of genes to be able to make statements about how as a particular preparation is responding to some stimulus in the environment, and basically understanding what pathways are getting turned on or turned off in a cell in response to some stimulus. Well, he does that because all of these results are stored in something called the Gene Expression Omnibus, or GEO. GEO is a huge database maintained by the NIH that stores the result of every microarray experiment that has ever been done with federal funds in our country. And the interesting thing about GEO is that it stores information about all kinds of experiments, and by searching GEO for experiments, people can find out what other people have done in certain areas of science. This is becoming the enterprise not of other people's money, but of other people's data. And the idea here is that investigators are able to look online at GEO, look at what other people have done in certain terms of other experiments, and people like my friend Pravesh Khatri can do remarkable things. Pravesh has done studies where he's been able to look online in GEO and identify what genes get turned on when a patient has sepsis. He basically has a biomarker in terms of a set of genes that get turned on that allows doctors to know when a patient is septic. He has a biomarker for TB, knowing what genes get turned on when somebody has TB, and now WHO is working all over the world to use this biomarker as a way of identifying patients who have TB, when previously tests for TB would take weeks to know whether they were positive or negative. Here, Pravesh can do this in an afternoon. Pravesh has done studies looking at distinguishing viral infection from bacterial infection. He's looked at ways of identifying when an organ is being rejected. He's done all these great things, made all these discoveries, and he has never walked into a lab. He's never put on a white coat. He's never used a pipette. He's been successful because he has been able to examine other people's data and use other people's data to make really great, remarkable discoveries. There's so much interest in this kind of work that there is a conference called the Pacific Symposium on Biocomputing, which actually gives an award each year for the best data parasites. And this is really something you should aspire to participate in. If you take other people's data, make observations that lead to discoveries that are biologically interesting, then you too can get a data parasite award from the PSB conference. Pravesh has actually won hands down in the first round, and this is something that's offered every year, and people get really excited about the idea of being data parasites, but there's a problem, and you already know what it is. Most of the data out there that data parasites want to analyze are terrible. The data that are out there in public repositories need to be findable through some sort of a search facility. So those search facilities may or may not help us. They need to be retrievable in a standard format. Often we don't even have standards, let alone standard formats. They need to be self-describing so third parties can understand what was actually done in order to perform these experiments. They have to be the product of careful planning and organization and stewardship. And the data themselves can't just be thrown up online. They have to be put there with the intention that someone is going to reuse them. And unfortunately, the culture of science is not one that thinks about data as something to be put online for the benefit of others. Unfortunately, most scientists view data being put online as something that they need to do to tick a box so they can get more funding from the EU or they get more funding from the NIH, but not really thinking about that in terms of what it means to get funding because we're doing something for the greater good. Well, the real issue is people got together and said, we can't continue to have this kind of a situation. We have to recognize that our data parasites need to be able to have access to data in all possible ways to be able to make successful analyses of such data. About 10 years ago, a group of people got together in Leiden and said, look, we don't want to just put data online. We want data to be online in a way that we call FAIR. FAIR has become this big acronym that in the scientific community has just been everywhere. FAIR stands for findable, accessible, interoperable, and reusable. And the idea is that if data satisfy those principles, then data parasites like my friend Pravesh Khatri can get access to the data, do these experiments, and do all kinds of great stuff, making new discoveries. And when you look at what's going on in the natural sciences, it's unbelievable. Like everybody says, data has to be fair. My data have to be fairer than your data. My data, in fact, are fairer than your data. And it almost is like it doesn't matter whether my data are correct or my data are true. What matters is my data are fair. Well, the problem is making data fair is hard. The people who articulated the idea of fair data said, if your data will be fair if you follow these 15 principles. The problem is nobody remembers what those 15 principles are, so please don't try to memorize them now. You can't. But the important thing to take away is when you look at the FAIR principles, almost all of them deal with metadata. A lot of them deal with things that have to do with the repository where the data may be stored. We don't really care about that so much because most investigators don't have any direct control over how the repository is managed. But when you look at what it says about the metadata, they have to be rich, they have to use vocabularies that themselves are fair, they have to be richly described, they have to be they have to use domain-relevant community standards. These are things that are obviously hard to determine computationally. These are things that are highly subjective. But it drives home the fact that if we want to have fair data, if we want to make our friends like Pravesh Khatri succeed at being data parasites, we need to do something about our metadata to make them more interesting, more useful, and more searchable. That's really important because when you look online and you see what metadata look like, they're terrible. It turns out they're terrible because investigators view their goal as to publish papers. They want to generate PDF documents. That's how they get tenure. That's how they get promoted. They don't get promoted necessarily because they do a really good job of annotating data sets. Sponsors, encourage us to put data online to allow other people to access our data, but they don't necessarily pay for it very much. And frankly, creating good metadata is really hard. It's hard because most of the time scientists create their metadata by filling out flow sheets like this one. And it's a nice spreadsheet, but the spreadsheet gives you very little guidance. You don't actually know what are the things you need to fill in. You don't know what the precise terms need to be if they come from controlled terminologies. Basically, what happens is when you create metadata using these kinds of undirected spreadsheets, you get stuff like this. This is an example of metadata that comes from the biosample repository at the National Center for Biotechnology Information at NIH. What do we see here? We see a bunch of attributes of information. We see things like cell line and cell culture and repository and tissue and disease. And actually, that's not so bad. We can kind of make sense. We kind of know what those mean. But what does it mean to say that our culture collection is GNE:586138? Most of us don't have a real intuitive feeling about what that means. What does it mean to say that the tissue is liver? Well, we kind of know what the liver is. That the disease is carcinoma hepatocellular. Well, that's not a term you'll find in any dictionary. Now, hepatocellular carcinoma you might find in a dictionary, but this inversion of carcinoma hepatocellular, not so much. Ethnicity, Japanese? Is that really an ethnicity? Could be. Age, 57 year. Well, if you're searching for the age of something, you might say 57 years plural, but not 57 years singular. This obviously is not completely what you would want if you want to be able to search for information online and get what you need. But it gets worse. Suppose we have this, and yes, I won't say this is a totally randomly selected sample, but here we have cell type, primary cell of what? Ethnicity, Asian, health state, SLE, You've got to know what SLE means in order to know what health state means. And as SLE, which I assume is systemic lupus erythematosus, a disease, really the same thing as a health state. Again, we really don't know. Race, yellow race, what the hell is that? In other words, there are all kinds of metadata that people create, putting them online, and if you want to find this record, you have to know what to search for, and you don't necessarily know that you need to search for something as weird as some of these entries. Then you see stuff like missing colon, missing pop, tissue liver disease, HCC. I think that's another variant on hepatocellular carcinoma, but don't quote me on that. Here's another example where, again, where the attributes are all over the place, the values are all over the place. Notice sex W. I'm not sure what sex W is, but we can make some guesses. I mean, this is the kind of stuff that we see all over the online repositories that scientists are supposed to use in order to do secondary analysis of data and make sense of them. You ask, how is Pravesh Khatri so successful in the work that he does? He's successful not because he has great technology, because he has great insight, because he's very smart, but he is very smart. He's successful because he has about 30 graduate students who spend inordinate amounts of time manually going through the records of these databases, trying to figure out what the authors of the data entries must have meant when they do stupid stuff like saying altitude C. I mean, no one knows what that means, but he asks his graduate students to try to figure that out in an effort to try to understand what could be going on. We have looked at very comprehensive analyses of databases where people put their stuff online. This is from Biosample, one of the other NCBI databases at NIH. When you ask a question of the metadata in the database, of those metadata that are referring to Boolean values, how many of the Boolean values are actually true or false? Well, it's about 73%. 73% are not true or false. What are they? Obvious things like non-smoker, former smoker. You ask, how many metadata values that are supposed to be integers are representable as numbers? Well, only about 26%. You can see other things which are, I don't know if those are numbers or not, but they're certainly not the kinds of things that I would expect in this kind of a repository. 68% of metadata entries that are supposed to represent terms from biological ontologies are not terms from biological ontologies. There's stuff like presumed normal wild. Basically, there's stuff that if you were searching for them, you would never find them. What does that mean? It means that if we want to be able to have fair data, and of course everybody is already claiming that their data are fair, but if we really want fair data, we want to be able to have a mechanism by which we can make these data sets accessible and viewable and searchable because we're using metadata entries that are consistent. So we want ontologies that give us the control terms that we need, that will ensure that we know exactly what we're talking about. We want reporting guidelines that tell us what are the attributes of experiments, the attributes of the samples, the attributes of the experimental conditions that will allow us to know very clearly what is it that the experimenter has done so that we can search for them and get the data fairly. We need technology. Of course, most of us are in computer science, so it's always good to talk about technology. We need technology that makes it easy to author good metadata in the first place, and we need procedures that allow us to be able to create the necessary standards, to apply the necessary standards. Basically, there's a whole cultural component, which I don't want to, I list last, but I don't want to suggest that it's not important. It's critically important because most of the time, we have to deal with the creation of data sets where the necessary standards that would allow us to search for the data and find the data in a fair way don't exist at all. These are the kinds of things that we need, and let's talk about these one by one. Let's start with ontologies. Here's GEO, the Gene Expression Omnibus, the database that Pravesh Khatri searches all the time. If you want to find a subject of a certain number of years, a certain age, well, you get to search for age and age and age and pop age and age after birth and a million different ways of saying age. If you're searching for age, you or those 50 graduate students you've hired have to recognize all the different ways in which somebody can say the word age, and you have to anticipate all those differences. You have to anticipate all the possible typos. You have to recognize that because no one uses ontologies in geo, you can't find easily the things that you want. We at Stanford have created a resource called BioPortal. BioPortal is a repository of all of the world's publicly available biomedical ontologies. And you can see some of the more commonly used ontologies like SNOMED CT, which is used in clinical care, MEDRA, which the Food and Drug Administration in the US uses to report adverse drug events, CPT, which is used to report interventions that are done on patients in the clinic, RxNorm, information about drugs, and millions and millions, not millions and millions, but 1,000 ontologies that people have created in biomedicine that are all accessible through BioPortal, and most important, that are all accessible through the same API. So if someone wants to find information about an ontology, then BioPortal is a resource that makes it very easy to do so, and that's one piece of the puzzle to try to make our data more fair. BioPortal allows us to look at individual ontologies. Here's something called the foundational model of anatomy, which is, as you would guess, a description of organs and their relationships. And we can see all the organs that have been enumerated in the system for this particular ontology. This is a way of talking about the organs in the body with extreme precision in ways which are not possible if you just give somebody a spreadsheet and say, fill in this blank. But we need more than just ontologies. We need reporting guidelines. So the reporting guideline is the list of attributes of the experiment that allow you to be sure that you've said what you need to say about the experiment so that people can find your description of the experiment and so a third party can know exactly what was done. So here's a microarray. These are the technologies. Actually, they're kind of going out of favor now, but they're very, very important in biology. They basically allow you to have genes placed on this matrix. The genes get turned on and turned off, and then they glow. The fluorescent dye glows when they're turned on. You can see over time which genes are activated, which genes are inactivated. And in order to describe such an experiment, the people who work in this area of biology said, look, we can't just describe our experiments in prose any way we'd like. We have to be very explicit about what was the substrate of the experiment, what was the platform, the micro system that we used, what were the experimental conditions. And what biologists did is get together about 20 years ago, and they created a standard called Miami. Miami is the minimal information about a microarray experiment. One of my favorite acronyms. And Miami was one of the first examples of how biologists as a community got together and worked together, not because anybody was imposing this on them, to describe what are the things about a microarray experiment that you need to specify in order for someone to make sense of what you've done. And Miami became incredibly important, and it became the first step toward a standardization process for describing data sets in a very coherent, reproducible way. So we had Miami for microarray experiments, and soon we had MIATA and Miriam and MiniMES and Miss Fishy and all kinds of really great acronyms, which actually have started a trend of trying to be able to describe for the biomedical community what are the terms that need to be included to describe an experiment in order for someone to make sense of it. And so we had suddenly all of these reporting guidelines coming on board, ontologies coming on board, but our metadata still were kind of messy. You saw what they look like. How do we actually get people to apply such guidelines and apply such ontologies in a useful way? We recognize that those ontologies are critical for describing the values. The reporting guidelines are critical for describing the attributes. What we really need is technology that makes it easy to author metadata using guidelines and ontologies. And that's where CEDAR comes in. So CEDAR, which I'll talk about first, is a workbench that allows investigators to use reporting guidelines and ontologies to edit their descriptions of metadata so you don't end up with sex W and other kinds of bizarre stuff as we saw earlier. I'll also talk a little bit about the CEDAR metadata validator, which is another approach we're using, again, to make better metadata, to make a more clear description of how we can describe online what our data sets are about. So here's sort of like a flow diagram as a cartoon of what the CEDAR workbench looks like. What you see in the left-hand box is that you have a mechanism whereby authors can create templates that describe the reporting guidelines for different classes of experiments. And so for each template that you create, you're defining what are the things about some class of experiment that you want to include. What are the data types for those things if they are actually going to be described using terms from ontologies, then you want to say what are the ontology terms, or what is the branch of the ontology or the whole ontology that you might use to provide values for those particular attributes. And so you have an easy way of defining templates that will indicate the attribute value pairs that define a particular kind of metadata guideline. Not the specific values, but the kinds of values, so you can ultimately use the CEDAR mechanism to fill that all in. So here are what a bunch of templates might look like in the CEDAR library. Suppose we're interested in biosample, one of the NCBI repositories. Biosample has a reporting guideline saying what should biosamples indicate so that you know what the biosample is. We say we're going to select it, we're going to select populate, and here we see the elements of the reporting guideline, the fields that need to be filled in order to create a description of a sample in a structured way. And then the question becomes, well, what are the values for those fields that are going to that you need to specify for a particular instance of such a reporting guideline? So we might say the sample name is 056, the organism is Homo sapiens, the tissue is, well, here's what you get a drop-down list. And it's not any old drop-down list. Remember, CEDAR stores in a cache all of the metadata that it creates. So we assume that those metadata represent community standards, and we can learn from those metadata what are reasonable patterns in the metadata that might be appropriate when we want to author future metadata. So in the case of the tissue, in the case of biosample, we know from our cache that if we're dealing with human subjects, most of the time we're dealing with blood as our tissue type. And next to that, liver is probably the next most commonly selected tissue type. And after that comes bone marrow and so on. And not only are these just the most commonly used entries, but we can see very specifically, these are terms that are taken from the Uberon ontology, which is an ontology of anatomy that includes a very rich description of human tissues. And so we're not just selecting any old term, we're selecting tissue terms from Uberon, and the menu happens to be sorted in a way which makes it really easy, because the sorting of the menu is done in a way which puts basically at the top the most frequently selected entries based on our previously entered values into this form. Now, suppose I'm filling in the same form. I've said that the organism is Homo sapiens and the tissue is lung. When I get down to filling in the value for disease, you can see that if the tissue is lung, I get things on my drop-down like lung cancer and COPD and squamous cell carcinoma of the lung and things that you find in your lung. Because again, we're able to go to the cache of previously entered metadata and sort the drop-down list, so the most commonly selected stuff is at the top. If I say that our tissue is brain, then I'm not going to get lung stuff at the top. I'm going to get brain stuff at the top. I'm going to get Parkinson's disease and CNS lymphoma and autistic disorder and melanoma of the brain and Edwards syndrome and schizophrenia and so on. And so we do our best to make it easy to fill in these forms because we recognize that this is hard. Investigators would much rather fill in that silly old spreadsheet I showed you a few minutes ago than actually have to go through the care of making the right selections to make sure that they're using the right reporting guideline and the right ontology terms. But if we can do it in a way where the terms are presented in an easily selectable form, where the amount of clicking is kept to a minimum, the hope is that we'll make life simple. And we've been very excited by the way in which CEDAR has been picked up by a number of very large consortia, generally funded by the NIH. Consortia like HubMap, which is trying to map every biomarker in the human body, HEAL, which is looking at the problem of opioid addiction in the United States, RADx, which is a project that I'm involved in, looking at measurements of diagnostic efficiency for patients who have COVID disease. MCBK is actually looking at ways of packaging up biomedical knowledge. So a lot of big projects, all of which are trying to create metadata in a way which will make the corresponding digital objects that are a result of all these experiments fair, findable, accessible, interoperable, and reusable. What's really, really exciting for us is that not only are these big consortia picking up CEDAR, but there are a bunch of publicly available, what are called generalist data repositories that are recognizing that they have metadata problems as well. This is Dryad. Dryad is one of several what we call generalist data repositories, not necessarily tailored to any particular kind of investigation, but used widely in science as a way of storing information about scientific experiments. And Dryad is now working with us to incorporate CEDAR so that not only will people put their data in Dryad, they'll put their metadata in Dryad, and those metadata will not just be trivial metadata, they'll be rich metadata compliant with community standards. And the goal is by incorporating CEDAR into Dryad, those entries will be fair, and scientists will be able to use Dryad as a way of searching for data sets to do new experiments and make new discoveries. That's the hope. The Open Science Framework, another generalist repository, is doing the same thing, and we're getting really excited about this kind of work. Now, I mentioned that many large consortia are using CEDAR. One I'll talk about in a moment uses not the CEDAR system I just showed you using the web forms to fill out the metadata templates, but rather uses something that we call the metadata validator, because this is being used principally by HubMap, which is a large NIH consortium which loves what we're doing but says we hate web forms. They don't like filling in web forms. I guess it reminds them of doing their taxes. I don't know. But they've decided that what they really want to do is to use spreadsheets, because that's what they're familiar with, but do so in a more intelligent way. We gave them this beautiful web form for entering information about biomarkers, and they rejected it roundly. They say, what we want are spreadsheets that allow us to provide this kind of information. So what we've done for HubMap, and now we're experimenting with other groups, is to generate programmatically a spreadsheet whose headings come from the same elements as the reporting guideline that you enter into the CEDAR template. So the idea is we're kind of meeting our customer halfway, saying, look, we won't give you an empty spreadsheet. We'll give you a spreadsheet where the headings represent the elements of the reporting guideline. And actually, if we know that one of those headings needs to be filled in with terms from an ontology, we'll actually bury into the spreadsheet. You can't see it, but when you get a drop-down from the appropriate cell, you'll get a list of terms that correspond to the ontology terms that would correspond to that particular entry. And I'm embarrassed to say they absolutely love this. The challenge, of course, is that when you give someone a spreadsheet for data entry, you open up the possibility that all kinds of things can go wrong, because you give them the ability to enter things which are outside of drop-down lists and outside of standardized mechanisms for ensuring correctness. So what we've developed is something which we call the spreadsheet validator, which allows the user to take that spreadsheet after it's been filled out, drag it into the box, and what the spreadsheet validator will do is to look at the spreadsheet, look at the template as it exists in CEDAR, and say, okay, you've done a good job filling in all the blanks, but even though you've tried to do a really good job, there's still some serious problems, and some of your metadata are invalid. And it will tell you, for example, that some of the metadata values that should be there are missing. You just didn't fill them into the spreadsheet. Some of the metadata values that you've entered are just plain wrong. They don't adhere to the ontologies. They don't make any sense given what we know about what's expected of those values. And in fact, we can help you. We can show you how to correct the values that are incorrect to make them compliant with what those values should be if you were using the CEDAR template correctly. And so what these folks are doing, and they find this much more tolerable than creating everything de novo in a web form, is to fill out their spreadsheet, run it through the validator. Validator will help correct all the errors, and the validator will then upload the metadata to the data coordinating center so that the correct metadata are preserved and everything is okay, and everybody is happy because they never had to fill out those web forms in the first place. I'm obviously surprised how successful this has been, because I didn't think it was going to be successful at all. But it does emphasize the importance of being able to accommodate the ways in which users are familiar with filling out, are familiar with accomplishing their tasks, and the need to be able to use the paradigms that users find familiar whenever possible. Because obviously this is something that they understand, whereas the web forms were not. So we talked about two kinds of technology. We talked about the CEDAR Workbench, which allows you to create templates that are compliant with reporting guidelines using appropriate ontologies, giving you a web form to fill out. This CEDAR Metadata Validator, which does basically the same thing, but has the intermediate step of allowing you to fill out a spreadsheet, which then gets validated. And we're really very excited that these are now being taken up by these groups I've mentioned, and gives us an opportunity to experiment with how well this kind of technology can enhance the ability of scientists to create data, which not only is going to be annotated with metadata that will go online, but with metadata that what people consider to be fair and will allow the data sets themselves to be fair. That's all really nice to say, but I've kind of glossed over the fact that we still need to have templates. We still need to have standards. We still need to have ontologies. And the FAIR principles not only require all that, they require that the metadata be rich, whatever that means. They require that that metadata adhere to relevant community standards, whatever that really means. How do we get those? Where do they come from? I can't just go to a book and find relevant community standards for an arbitrary set of experiments. Basically, I have to create the mechanism by which those community standards get encoded and preserved for use by the computer. And my colleagues at the GoFair Foundation have been doing this for the past 5 years in a way which has been very, very successful. Well, their procedures have been successful. I will say less about their logo, but in any event, the GoFair folks create what they call metadata for machine workshops, or M4Ms. And in these metadata for machine workshops, what they do is they get together with a group of domain specialists who are handpicked because they presumably represent the best scientists in a particular discipline. They put them in a room for 2 to 3 days. I think they're allowed out occasionally. And they work with them to identify what do these scientists believe are the essential things that need to be said about an experiment for a Pravesh Khatri to figure out what you've done. And these M4M workshops ultimately will end up with something like Miami, but for a different area of investigation. So the M4M workshops are able to lead to the description of metadata templates that are designed as standards for new areas of science. And it's a model of working with experts, identifying how they think about problems, using that interaction as a basis for coming up with a representation, which ultimately is encoded for the computer, in our case, in terms of a CEDAR template and sometimes in terms of new ontologies. It's interesting because this has been picked up very quickly in Holland by the Dutch Agency for Health Research and Development, or ZONMV, which means that in Dutch. ZONMV says, we want our data to be fair. We recognize that data can only be fair if we have appropriate community-based standards, and investigators use those standards. What the Dutch do is really interesting, though. They don't just say, go out and find some standards, because they know the standards often just don't exist. What the Dutch do is they contract with the Go Fair Foundation, and they will run a metadata for machines workshop in advance of issuing an RFA for a new area of science. And they will use that M4M workshop to articulate a new standard, generally in the area of something related to COVID, because that's where most of the work has been done. And then they will say, if you want our money, if you want to apply for this RFA, then you must promise that you will collect data, and you will collect data whose metadata will adhere to the standard that came out of the M4M workshop. It's the funder saying, we believe so much in fair data and open standards that we're going to write into the RFA a requirement that if you take the funding, you must deliver your data in an open format that adheres to this particular standard for metadata. It's I think it's remarkable. My colleagues at NIH say it's draconian. But in any event, what it's leading to is an increase in the amount of data available online in the Netherlands that would adhere to the FAIR standards and that people are able to use for new experiments, for secondary analysis, and hopefully new discoveries. It's really quite interesting. And it's really a model for how funding agencies who care about data fairness can make demands of investigators so that those investigators will do work in a way which generates fair data. Now, the Dutch mechanism here, of course, is rather strong. It's basically telling scientists, thou shalt. And that doesn't always go over very well. As I said, my colleagues in the United States are very nervous about making those kinds of demands. But I think, frankly, that's the kind of stick that's going to be needed to complement our existing carrots if we want to be able to see investigators really generate data that are fair and open and useful to other people. So basically, when you think about it, what we've been talking about this afternoon is CEDAR and the CEDAR validator and the whole mechanism of trying to make data fair. And we've been talking about this in terms of guidelines, we've been talking about it in terms of technology, we've been talking about it in terms of these computer programs and spreadsheets. But that's not exactly what's interesting here. For me, at least, what's interesting here is that everything that we're doing in the CEDAR approach requires modeling. We have to create models of guidelines, of these reporting guidelines that tell us what to say about experiments for those statements about the experiments to be meaningful to other investigators. We're making models of ontologies that tell us how do we model the world to create the defined and precise terms that are needed in order to be able to describe experiments so that other investigators and third parties can get access to the data that they want. And we're modeling workflows for data annotation, for data stewardship, so that we can build the kinds of systems that will fit into the kind of flow that is necessary for investigators to be able to describe their work in a way that is familiar and easy and allows us ultimately to have not just data online, but to have fair data where the metadata are sufficiently rich and sufficiently standardized that third parties can make sense of them. And I think one of the things which certainly is a common denominator in my work, and I sense to a great degree a common denominator of work that goes on in SES, is this notion of modeling and why it's so central to the kind of work that all of us does. And I think what makes life interesting for me is that CEDAR templates, building CEDAR templates, is just like the kind of work that we did 40 years ago when we were first working on knowledge-based systems in AI. And I realize most of you weren't even around then, but it was a time when there was enormous excitement about being able to take what it is that an expert knows and to render it into a machine processable form so that it could be reused over and over again in different ways. And I think that knowledge engineering activity, which was so common in AI at the time, is really what we need to do now as we work toward open science. That our goals now are just like they were at the time we were building knowledge-based systems. We're trying to be able to capture the knowledge of experts and put it into a machine renderable representation. And we're not going as far as trying to capture everything that experts do or know about some area and in some detail. We're not trying to build more Musen systems, more R1 systems, more, none of those big fancy expert systems, most of which were probably unable to meet their needs because they tried to do too much. But what we're trying to do here is actually much more self-contained and much more doable and much more valuable, I think. We're going to help communities of practice codify what they know about experiments in a reproducible way, enumerating the attributes of those experiments and the kinds of values that we need in order to describe those experiments. And when that happens, we will have not whole knowledge bases for solving whole problems, but we'll have the ability to plug and play with a variety of software systems. So right now, we can plug and play with the Cedar metadata editor, which uses web forms, or the Cedar spreadsheet validator, which uses spreadsheets, or other kinds of technologies that we'll all develop to make data stewardship much more automated and much more reasonable. And I think what's really exciting is that we will be in a position where we can work with experts to codify their knowledge of their experiments, to build these kinds of representations so that we can allow communities and people from outside the community to be able to reuse knowledge about those experiments in ways where there'll be absolute certainty about what it is that we need to know in order to understand an experiment, how we can look at what someone has done to process data to do an experiment, and we can use these kinds of templates over and over again to be able to know how a particular community approaches its work in science and how we can take the results of its scientific experimentation and reanalyze that information for new information to make new discoveries and ultimately to spawn a whole generation of people like Pravesh Khatri, who will never put on a white coat, who will never use a stethoscope, who will never learn how to pipette, but who will have data that they can use, data that will be valuable to them in order to make discoveries that no one else can make, and basically turn science into an enterprise that makes it possible to make new discoveries on the basis of work that other people have done, the same way that we can learn from others in many other disciplines that are not quite so siloed and where we don't have quite so many trade secrets. But that means not only being able to create better technology for making this all possible, doing better modeling in order to understand better what it is that we have to put into these reporting guidelines and ontologies. It also requires sociology of science, to understand where are the barriers, where are the hard parts, why is there so much resistance on the part of investigators to doing what we all think should be obvious if our goal is to generate new knowledge and to make that knowledge as accessible to everybody as possible. Let me stop there and be happy to answer any questions.